Still see me, or do you just see the PowerPoint?
Yeah, I see you.
Okay.
We're gonna start now. Well, hello everyone. Thank you for joining us in this seminar series. Today we are very pleased to have Dr. Carole-Anne Gillis. Dr. Gillis is a biologist with 14 years of experience working in aquatic ecology in rivers and lakes in 3 different countries, Canada, New Zealand, and the United States. She has a PhD in freshwater ecology, studying the impacts of Bibimosphenia geminata, a single-cell algae, on juvenile Atlantic cod. In 2014, Dr. Gillis took a role of director at the Restigouche River Watershed Management Council. The position was that of a cost-share fisheries biologist between local and indigenous groups. Constantly adapting and learning, Dr. Gillis works to strengthen the connections between traditional knowledge and Western science. She accepted the position of researcher director of the EPS Mi'kmaq Resource Council in 2018, where she continues to develop community-based research monitoring and restoration efforts at the watershed and storage scales. Thank you, Dr. Gillis, for being here with us. And again, sorry for my terrible pronunciation.
It's all good. Thank you, Andres. So hi, everybody. Today, I really wanted to present how fisheries research science looks outside academia and what can be done as a career path and open up options for your future uh, postgraduate studies and work. So I'm a biologist. I'm a research director at the Gispiga Resource Council. And I really, really love to promote women and girls in science. And we do a lot of work locally with new emerging and early career scientists. So that's something that I strongly believe in. And the path to success is, a bit different. So you always think that it's a straight shot. You do your college, your university, and then you find a job, and then that's a straight path to whatever you want to accomplish in life. But it really looks like, and especially this year, I hope that everybody figured that out, that it's quite of a mess to get where we want to go. And there's ups and downs and twists and turns. And yeah, so just keep looking forward and aiming for your dream job or your objectives, and you'll get there, but it's going to be a twisty, windy road. So really, as a graduate student and kind of wondering, like, what am I doing? What's this all for? What really motivates you? Why did you accept a position as a master, as a PhD, or a postdoc? What's the research that you're doing, and why does it motivate you? So that's a key factor in what you're doing, and even in your future when you're going to have a job. What motivates you? What is your why? So why are you doing all of this? So it's some big questions that it takes a long time to answer. You don't have to know what is your why, but you still have to sit and wonder and ponder, why am I working so hard towards this? So really, once you've identified that, it's easier to keep motivated. So when you're choosing your career, you're in high school, you're choosing your master's degree, your PhD project, your postdoc, your job. Are you aiming for a career? Are you aiming for a paycheck? Are you aiming for a specific position? Are you aiming for that prof position in a university? So really, you have to think about what are your values, what's your visions, what's your interest. I find that it's easy to be motivated once you're really, really interested in a topic. So throughout my path of education and work, the more I got towards more specific, so when it was just broad science, it was interesting. But when I really got into ecology and then fisheries ecology, I became more and more motivated and better at it because I was motivated and interested. So it was easier to study and do some research. I didn't know why I was motivated, and at first I was like, why am I interested, or what should I be interested in? What's my career path? What topic should I study? And I didn't really know what was interesting. So I liked science, but I didn't know what was going to be my path. So I had this friend, Rebecca. She was studying, she was a fisheries technician, and she was so passionate about what she was doing. It made me want to be passionate about something as well. So she would tell me all these stories about beavers and ecology of fish. And I was like, wow, this is amazing. So I went into biology and then figured out that, I liked fisheries ecology. This is a graphic depiction of the watershed that I belong to. So it's the Mattapitty River watershed that flows into the Restigouche River watershed. It's mostly habited upstream, which is a bit different from other watersheds in the world where settlers have established themselves at the confluences and near waterways and coastal areas. But then I became interested mostly in being home and studying something that impacted the watershed where I grew up. So this is Didymo. It's a single cell algae. It's a diatom that produces thick stock material. So all of this is just carbohydrates that single cells produced. So Dynamis geminata is a diatom. So as you see here, there's the cells. And at the pore field of the cell, they secrete these mucilaginous stalks and they divide themselves that way. So in poor nutrient stream, so when there's no phosphorus or nitrogen, this algae will produce stock material. So it's a bit counterintuitive, but it impacts rivers and streams in our area. So it all started in 2006 when fishermen discovered Didymo, and then they realized that it was here at first. Thought it was a invasive species because it was acting like an invasive species, and we didn't know where it was before or why it was here. There's a lot of work that was done in New Zealand 2 years prior, and it's been declared an invasive species in New Zealand. So we didn't know much information at all about this algae, and there's no studies that were done on the effects of it on Atlantic salmon. So the increase in prevalence of this algae in the watershed, and not only the Restigouche watershed, but everywhere else worldwide, was kind of a concern for communities that were living along the rivers and sustaining themselves with the Atlantic salmon industry and the Atlantic salmon as a food source. So my PhD really focused on controlling factors of mat formation in Didymo, the impact to the ecosystem, especially in regards to Atlantic salmon, and what caused it to be so disruptive and blooming so heavily in this watershed. So I really started working heavily in aquatic research and throughout my thesis with the watershed groups locally in the Restigouche watershed. Doing outreach and education with youth and sampling away at Didymo throughout my thesis. So then I realized what really motivated me. Was it the rock snot itself? Was it my topic of my study? Was it this brown fibrous mat of guck that was motivating me? Not really. So I realized that I was really motivated because it was so close to home. It was something that was impacting my river. So that's really where I was like, okay, this is why I'm motivated, and this is why I'm going to keep going and study. And then I realized I really wanted to do research in the long term in this watershed. And then I came to realize that it was because it was a sense of place. So that is my motivation for my career. It's all about community, family, relationships, landscape, your identity, your values, your experiences, your memories. So this is where I grew up. This is the Restigouche River watershed. And this is where I grew up, right here in the middle. This is where we're located. So this is the Gaspé Peninsula. This is New Brunswick, and this is Quebec. So right on the border of New Brunswick and Quebec, half of the watershed is in Quebec and the other half of the watershed is in New Brunswick, which is quite problematic when there's the concept of Atlantic bubbles right now with the pandemic. So half of our work is on the Quebec side. So I am a settler, Scottish and Irish and English and French Canadian, so a bit of everything. And so my experiences are from a settler point of view. So growing up in the fly fishing industry, 8th generation Atlantic salmon guide on the Restigouche, learning to fish, fly fish, being on the water, and then bringing that lens into research and bringing that way of knowing and being on the water to research. So observing the river my entire life and then actually realizing, hey, there's invertebrates, there's algae, there's whole ecosystems underneath this river. And so that was really, really interesting for me. And it's quite exciting to do, research on rivers and marine ecosystems. We're always out sampling with different methods. And then what really appealed to me to research, even though I had figured out my why, was really to travel. So right now it's kind of hard to travel, but we'll get there. We'll get there again, and hopefully you'll be able to do some internships with different researchers worldwide in the future. So I was able, thanks to Didimo, really, to travel pretty much everywhere in Europe and Northern Europe and New Zealand. I also went to Chile on the West Coast as well. So Didimo is found a bit everywhere in the Northern Hemisphere and has been considered invasive in the Southern Hemisphere. So I went out west to the Kananaskis River Valley and studied Didimo there. New Zealand, studied Didimo there as well. And then went to South Dakota and studied Didimo there in the Grand Teton National Park in Wyoming. Went to Chile and studied Didimo there as well. And then I came back to this watershed after traveling and doing some research, and I finished my PhD. And this is the depiction of my research area, of where I wanted to work and continue to do aquatic research. So in 2014, when my NSERC ran out, so I wasn't done my PhD, but I needed a job. So locally, we identified that there was a need to create some capacity building opportunities, both for the local watershed organizations, the local salmon management organizations, and also the First Nation communities, so the Mi'kmaq communities in the area. So mainly with Restigouche First Nation. So this is the view from our office. So this is on the Quebec side. The river is the boundary between the Quebec and New Brunswick provinces. And this is Campbellton. So this is Sugarloaf Mountain. And this is New Brunswick on the other side. So then coming back to the watershed where I was living and then seeing in the area, it wasn't just the Restigouche River watershed. You know, I was in Mi'kma'ki. I was working in Gisbigawagi. So it was really eye-opening for me to have grown up so close to the communities and not really knowing much about them. So Gisbigawagi is the 7th district of the Mi'kma'ki territory. Our offices are based in Restigouche. And GMRC, we work for the communities. We are an AROM group funded to conduct research on freshwater and oceans for 3 communities: Restigouche, Ugpiganjik, which is in New Brunswick, and Papineau First Nation. So really my job is to do some training, some capacity building, really work towards creating research opportunities here and answer key questions that are of concern of our member communities. So collect scientific data that are very useful for different issues that are happening in the watershed, and also looking at species at risk that are also very culturally important. So we do all kinds of activities throughout the watershed and coastal areas. We do electrofishing. We map out lake, aquatic plants, substrates, shorelines. We also use technology to do a lot of remote sensing work. So we collaborate a lot with universities. And so we mapped out the thermal refuges at the watershed scale. So with a helicopter, mounted a thermal camera and a high optical resolution camera to fly over all of the rivers in the watershed and map out the thermal anomalies. So this is an example of a picture that we would grab from the camera mounted on the helicopter. And then this is the associated thermal imagery. So the lighter the color, so yellow, it's hot, and the darker the color is colder. So you see a main stem with its tributary that creates a cold water plume. Over 2011, 2012, 2013, more than 861 kilometers of river were assessed, and we identified over 1,800 thermal refuges at the watershed scale. So these are the rivers that we flew over different years. So here is the Quebec part of the watershed. Campbellton is over here. It's been in the news a lot if you watch CBC News. And so the coastal area of the Restigouche and Béchelaar is towards here in the east. So these are all the thermal refuges that were mapped, and they were all characterized by the type. So then we can use this information to actually map how Atlantic salmon are using these thermal refuges in periods of drought and high water temperatures. And then we map the temperature and the temperature profiles, the persistence and size of thermal refuges over time. We also do another type of remote sensing with drones. So at GMRC, we have some drone technology. And this is a depiction of a cormorant colony. So we were looking at predation of the cormorants on outmigrating Atlantic salmon smolts. So we were looking at their diet. And so this was a partnership with the INRS University, and we had a master's student involved with this project. So cormorants puke up pellets, and in there you can identify fish otoliths that were undigested. And typically a cormorant will puke up 1 pellet every 24 hours. So you can get a full depiction of the diet over the last 24 hours. That's what a pellet looks like. And so we sampled during the outmigration of Atlantic salmon smolts to see the overlap in the diet. And per week, we sampled a minimum of 60 pellets by visiting the colony. So in 2014 and 2015, we gathered different pellets and sorted them out with dishwashing soap to remove all the grease. And then we remained with the pellets, and then you pair them together. So each pair of otolith equals one fish. And then we were able to assess the diet of the cormorant colony during the period of the outmigration of Atlantic salmon smolts. These are some of the different species that we found in the Bay of Chaleur. So it's also a great indication of what type of species are also occurring at the same time, and different species that are found in the Bay of Chaleur. We also do some telemetry work. So we tag salmon smolts and salmon kelts. So kelts are salmon that have spawned at least once and will outmigrate in the spring to go recondition themselves in the Gulf of St. Lawrence. And if they're a repeat spawner, they might come back after spending some time in the Gulf, recondition and spawn as a consecutive spawner, or they can be an alternate where they'll go back to the ocean and recondition over here. We do some telemetry on smolts. So this is a smolt wheel. It's a trap that spins in the spring to catch outmigrating smolts. And it's quite the effort to deploy one of these. And then we go every morning and sample the trap and see what different types of species. So yes, we're targeting Atlantic salmon smolt, but we also catch eels in this trap. We process all the fish every morning and then do a mark recapture study. So we put floy tags on all of the smolts or a portion of them, and then we bring them upstream and try to recapture them to do some stock level models to determine the productivity of certain river reaches and see what production are we having for Atlantic salmon in the watershed. And then we do morphometric measurements. So we look at the fork length and the weight of the smolts that are migrating to get a condition factor and a proxy of how healthy the smolts are before they go out. We do acoustic telemetry. And sometimes we involve our local fishermen to help us catch the kelts as well. So we'll be resuming this project in the spring. And then to detect these fish, we have to deploy receivers. So we deploy receivers in the river system, the estuary, and the Bay of Chaleur. And then we deploy acoustic releases as well. So we have to go in the bay and deploy and retrieve the data off of the receivers. We do spawner counts as well. So in the fall, we go down the rivers in diving suits and canoes and assess stock levels and prior to spawning, so we can really see out of what was out migrating at the smolt stage, what actually came back to the river, and what will be the recruitment for the following years. So I presented a lot of research from the Western point of view in the sampling, but I also want to introduce to you the concept of two-eyed seeing. So this concept was developed by Dr. Albert Marshall, and I really encourage you to read about it. And also there's this brand new paper that just came out by Dr. Andrea Jane Reed from 2020, and she really details the process of two-eyed seeing and in collaboration with Dr. Albert Marshall. Two-eyed seeing is, and this is my interpretation, so there's different accepted definitions, but it's really looking with a Western eye and also at the same time looking with an Indigenous perspective, and then gathering the strength of both knowledge systems, interweaving them so we can have a more holistic view of a different problem or a threat to the ecosystem or a study, So really to encompass all the ways of knowing into your research. So we do this by also conducting local knowledge and Mi'kmaq ecological knowledge interviews. So we gather knowledge with harvesters, fishers, local people that have been involved on the land, knowledge that's been passed down over generations and generations of Mi'kmaq. So we then want to interweave this knowledge systems together and have a better picture of a situation. And it's not a step-by-step approach, and there's no one way of doing it. It's really just welcoming both knowledge systems and then trying to figure out how they can complement each other. We do salmon health monitoring as well in the community. So we sample scales, measurements of length and weight of salmon. We do egg sampling to assess the fecundity rates of the Atlantic salmon as well. And this is also knowledge that we gather in terms of Mi'kmaq ecological knowledge. Are the salmon smaller now? Are you catching more females or males? Have the sex ratios changed over time? What have you noticed over time that has changed about the salmon population? And most times the scientific data and the MEK data fits perfectly together. We do scale reading as well. So we find that in the Restigouche system, over time, salmon are younger and younger. So we used to have elder salmon, very old salmon that would be repeat spawners above 12, 13, 14 years. And now we rarely get, an 8, 9, 10-year-old salmon. We do sampling also for virus testing. So concerns from the community were different bacteria and viruses. Are the salmon safe to eat? Are there any threats to the salmon coming back? So we sampled in collaboration with the fishers. We sampled different tissues, so gills, heart, the gut system, the spleen, the kidneys. And then we sampled this for 3 years. And we collaborated with different researchers to assess the bacterial and the viral profiles of the salmon. And so we found, and we've published this paper in CGFA, in Facets, sorry. And so it was a molecular assessment of the different infectious agents from different populations of Atlantic salmon. So Restigouche salmon were part of this sampling effort. And it couldn't have happened without the collaboration of the Restigouche Mi'kmaq fishermen. Then we found that Saprolegna was quite the issue in the watershed. So this is a fungal infection that we do see around spawning time, so right around this time of the year. Saprolegna is a secondary infection, so salmon that come in weakened already from the viral and the bacteria that were found in the previous study. Then in the fall, they're weakened, their immune system is weakened. And in cold water rivers, Saprolegna is an outbreak that can occur. And so we document that as well. We do research on other species as well, striped bass, for example. So we also looked at their diet and if they were a predator of outmigrating smolts. So we did find some prey items and overlap between striped bass coming into the river to feed and outmigrating smolts going out to the ocean. We also do American eel research, and this is one of the most of our best example of two-eyed seeing research that we have at GMRC. So we did some telemetry work, and we did also some interviews with local knowledge holders. So the local harvesters told us that eel do congregate at the fishing locations. So fishers come in and sometimes clean their fish directly at the wharves, and the fishermen see the eels feed and swim around these areas. So our telemetry actually confirmed that. So it was really interesting to have the ecological knowledge on one end and then have the telemetry work and have them overlap perfectly. We also did some estuary health sampling, so different fish communities, macrophyte substrates in the area. We do fish identification. We also do Scotty boxes. So we do, as you see, I'm just going through all these pictures, and it's just the research is so diverse. But what's interesting about it is that it's place-based. And that's what's really interesting is you have these watersheds and this coastal area, and really you can do any research topic at all in this area because it's so place-based. So we do Scotty boxes as well. So this is incubators that we put in the river. We put green eggs in here, and then we assess survival rates the next following spring. We've also started working on Atlantic sturgeon. So we found that in the Restigouche River estuary, Mi'kmaq fishers were catching, not often, occasionally Atlantic sturgeon, and we wanted to know why that was happening. And so we actually found a critical nursery habitat in the Restigouche River watershed this summer when we sampled. So it was absolutely interesting to map out that we have this in this area. We also do electrofishing, and we assess culverts and fish passability issues. So we do that throughout the watersheds. We also do salmon summits. This year we didn't do a salmon summit yet due to meeting restrictions. So we really bring in all the salmon stakeholders and right holders in the watersheds and bring them together to discuss salmon issues. It's quite the interesting event of bringing different ways of knowing together and going about how to find actionable items to address Atlantic salmon conservation in the area. So I've been talking about the different awesome research that we do outside academia, but it doesn't mean that we're not partnering with academia. So it's really, really interesting to have a place-based research program that is pretty much open to community concerns and the key aquatic habitats that we have in this area. So the research is pretty much limitless. So it's really interesting to be part of this research program with local communities that where you're doing actually applied research that will impact decision making locally. So it's really a short connection between the impact of your research and the action items that can actually be taken. So a few take homes about what can motivate you and how to go towards where you want to go in your career. Just a few take homes. You're limitless, you're smart, you're enough. Hard work pays off. Find out what motivates you. What is your why? Create a vision and a goal for your career. Act with intent and get out of your comfort zone. Every time you will get out of your comfort zone, you will learn tremendously. And lastly, get yourself a Twitter account. You're a researcher, you're a science person, get yourself a Twitter account. Follow people and get inspired. Really, it's really an important thing, especially in fisheries research. It was one of the best recommendations I've ever received throughout my early career. And then realize, where are you standing right now? Whose territory are you on? Which community is nearby? What research are you doing, and how does that impact resources for different communities in your area? Recognize your bias, your colonized way of thinking. Recognize that you are a settler in Canada or wherever you're listening from. Don't assume anything about communities, Indigenous communities. Just come in with an open mind and don't assume anything. Learn about two-eyed seeing. Read about the work from Dr. Albert Marshall, and more recently, the work from Dr. Andrea Jane Reed. And actually, today, at 1 p.m. Newfoundland time, there's this event that I hope you can attend. There's still time to register. Different researchers, Indigenous academics will be presenting this concept. So it's going to be great. Go on waysofknowingforum.ca and sign up with Eventbrite. And it's a small contribution to attend the talk, and I really recommend it. And then in Newfoundland, get to know your local air-round body. So MAMCA is located in Newfoundland, and get to know them, get to know their work, and get to learn the communities that are in your area. So the Conn River and Corner Brook as well. So yeah, I hope I didn't talk too long. Do you have any questions? And feel free to ask me anything. Thank you.
Thank you very much, Dr. Gillis, for such an interesting presentation. If anyone has a question, you can unmute yourself and ask her.
Do I have to stop sharing my screen?
I'll ask first question. I was wondering, like, you mentioned at first that algae that you were working on, the problem with that is about the habitat for salmon. Of what is the problem that can cause for salmon, and how is the situation now?
So Didymo is a stock-forming diatom. So when it grows in streams and rivers, it's actually going to cover the entire riverbed. So hypoaric exchange changes, the invertebrate community changes, the behavior of the juvenile Atlantic salmon also changes. So you can imagine, juvenile Atlantic salmon, they have this foraging behavior where they have a very territorial approach. So they'll be on their home rock and they'll defend that little 1 meter square patch of habitat, and they'll feed on that home rock pretty much throughout the summer unless they're displaced. So in that portion of habitat, imagine it just being fully covered. So all the interstices, the spacing between the substrate is covered by this fibrous, thick 1-inch mat. So your prey is located in different areas. The way you're feeding is a bit altered. So instead of just waiting for drift to come downstream, you're actually foraging onto the mat. So bioenergetics are changed in Atlantic salmon, and we found that lipid content was actually lower in Didymo affected reaches than non-Didymo affected reaches.
And is there a way to control it somehow, or?
Not really. So it's a diatom that blooms when the nutrients are limiting. So when it's nutrient limited, it's actually going to produce the stock. So it's not that you can just reduce the phosphorus, because that's what promotes it. There's been some phosphorus input work that was done in South Dakota. It doesn't really work because you're going to produce a lot more phosphorus, you're going to input it, cell division is going to increase, and mat thickness is going to decrease. But then you're just causing a lot of cells that can just colonize downstream an area where there is phosphorus limitation. So you're just moving the issue downstream. So in different areas, streams are too oligotrophic, so they're very nutrient poor. And so really Didymo is almost like a sentinel of climate change indication in different rivers. And you can't really control it. So in New Zealand, where 90% of the rivers have a hydro dam, you can do flushing flows. So you can have events of scouring, but again, you have to have flushing flows that are so impressive and so high that you create this bedload transport in your substrate, so it can erode the mat. But if you're just creating a shear stress enough to clean the mat, you're just going to promote growth even more. So yeah, you can't really do anything about Didymo.
Interesting. Another question that I had is, you mentioned a lot like the work that you do in collaboration with the communities, in the local communities, but I was wondering like what kind of experience do you have like getting back those results to the community, or how do you do that?
Mm-hmm. That's one of the most challenging things, really. What I find very motivating is that the community identifies an issue or a concern, and then we can develop a research proposal and bring in a two-eyed seeing approach to that project and carry out the work. We gather the knowledge, both the Mi'kmaq ecological knowledge and then the Western knowledge. So we use technology, we do data processing and sampling, and then we bring the two together. And that space, that portion of the project takes a lot of time. So that's a part of the project that is very time consuming but necessary. So then when we come to the results of showcasing the work, so we can present, we can publish the work and then share it with the community, but really we try to do information sessions and lunch and learns with the community members to present the results that we do have. So we actually come back and interpret the results that we have and communicate that. So now we're kind of in this moment where we can't have that face-to-face lunch and learn community meeting style. So we're adapting right now in trying to find ways of communicating results with two community members. So we're at that point where we're trying to do webinars and share that. We're also doing Facebook Lives with the communities. So we try to share as much on social media as we can. But I'm looking forward to the time where we can go back and really sit down with the fishers. I find the time that we share the most is actually when we're at the wharf and just talking with community members about the research that we're doing and then what they're seeing. So really it's really the moment that we're both, on the land, on the water together, that we share the most together.
Thank you. Any other question for Dr. Gillis?
Dr. Gillis, I was just wondering if you could touch a little bit more on the trends that you've seen with like climate change that affects the growth of this algae.
So climate change effects on the growth of the algae?
Yeah, like what kind of things have you seen or you've noticed?
Okay. So worldwide, there's different hypotheses. So there's nitrogen deposition levels that have increased worldwide, and that's impacting stoichiometry and nutrient dynamics in rivers and lakes. And then what we've also found locally, we looked at the river ice out date and how that has changed over time. So we have data going back from 1894 to today. So we see now that overall the ice out is happening around 9 days earlier. So thermal regimes and ice persistent is changing over time. We're also seeing air temperature is being on average a bit higher. So we have about 3 degrees warmer on average for air temperature in the area, again on a pretty much a century database. We're also seeing that rivers are getting lower and lower. We're seeing that rivers are getting warmer. We're also seeing that there's a lot of flashiness. So high discharge levels, and then very, very low discharge. And then we usually see a spring discharge that's very high due to snowmelt, but sometimes we'll see 2 peaks because we get a lot of rain or the ice out is not timed as it used to be. So we're seeing a mismatch in the hydrological diagram for the rivers locally. So we're changing, that can all affect the persistence and severity of Didymo and also other biota in the watershed.
Interesting, thank you.
Thank you.
And I was also wondering, you mentioned that Didymo is based all over the world. So, and they are also an invasive species. So I was just wondering what are the means of getting from point A to point B for them? Like
the spread?
Yes.
Okay. So at first it was really, really hard to identify, is it an invasive species? Is it not? Is it exotic? Is it not? Is it disruptive? Is it not? So it was really, really hard to come to terms with it. We went through old observations. There was some earlier studies from the 1900s, very around the area, and that they had found Didymo cells, but it was so rare. It was not a species that was found broadly 100 years ago. When you found it, it was actually almost like a trophy because it was like, oh my God, that means this river is so clean. This river is oligotrophic. And it was an indicator, a bioindicator of pure, clean water. And then flash forward to the 2000s, it's been considered as a native species of the Northern Hemisphere. And but in the Southern Hemisphere, it's been considered as exotic. So it's native to North America and Northern Europe, but it's acting as an invasive species because the environmental conditions are making it completely proliferate and bloom and cover entire riverbeds completely. So we did some paleo work earlier on in 2012 to see, was it native from this watershed or not? So we went in the sediment of lakes and we did a core. So we were able to find it in our core. We can go back as far as we wanted, but we did find some sparse Didymo cells from the early 1970s. So that became the point where the Quebec government decided, well, this species is not exotic. So that's when they pulled the funding on the research because it wasn't invasive anymore. But it's still disruptive. So it's not exotic, but it's invasive due to environmental controls. So, and can it be spread? Well, it's, a microscopic species. It's been thought that fishermen and different users of the river have spread it. In Chile, they've actually found that minks can actually spread Didymo. So, yeah, it can be spread. The spread explain its current Northern Hemisphere distribution? Probably not. Does it explain its distribution in the Southern Hemisphere? Probably. So it's still all up in the air.
Thank you very much. It's very interesting.
Thank you.
Any other question?
You can also put questions in the chat if you don't want to unmute yourself.
Yeah, exactly. If not, it's just want to thank Dr. Gillis for being here and giving us that interesting presentation. And for all of you, thank you for joining us too, and stay tuned for the next seminars of the series.
Right. Thank you for having
Thank
me.
you very much.
Oh, I think we have a question. Ashley Cook, I was wondering if you could post the link. Oh, I think it's for you, Anders.
Okay.
Oh, that's for me. Sorry.